So our next speaker is Hervé Bocherens, who's professor at Tübingen University, and he's going to talk about Neanderthal palaeoecology, insight from stable isotopes. Thank you.
Thank you very much for this nice introduction, and thank you for the invitation to participate to this very interesting meeting. I must say I learned a lot. And now I'm going to talk about my topic, which is insights from stable isotopes on Neanderthal ecology. Thank you, Bruce, to have introduced the topic, even if I'm going to, to repeat some things, but also to modify some interpretations. I mean, the method has evolved and is going further. So I will start like you with pictures. So we have these ideas about the ecology of Neanderthals, with some of the images from Mauricio. So, Neanderthal hunting, it's a clear idea. I mean, what could they do else? And, but hunting where, hunting what? So hunting maybe deer, but hunting in a forest, or hunting in a open, cold landscape, and hunting big animals, hunting in groups, hunting alone. But they are also maybe fishing. This is also another possibility to get food. And they could also eat plants, of course. So these are all the possibilities that they have, and we want to try to figure out how much they eat of what and where did they do that. Another idea is that did they eat fresh or did they eat rotted things? Did they scavenge or did they hunt? This is also a debate that has been around for a while. So, my background is paleontology, not archaeology, and so I have a view of the world which is maybe a bit different. I see the world as an ecosystem, and so I see Neanderthals as part of the ecosystem. So I'm interested in seeing Neanderthals as consumers, so they are part of the ecosystem. They are also competitors with other species that are around. What is also important is to evaluate the flexibility of the Neanderthals through space, through time, and also, we cannot ignore that there is also a number of theories or explanations for the extinction and the replacement of Neanderthals by modern humans based on ecological scenario, like modern humans were more flexible in their diet, were including more diverse resources, while the Neanderthals were rigid and only eating one thing. And so, of course, if you have competition between two species, one is restricted and the other one is more flexible, the more flexible will win. So we can also evaluate this idea. So this is how I picture this does not work. How I picture the ecosystem. So you have at the basis, you have the plants, but not just plants, it's very diverse as well. You have wooded plants, you have grass, you have lichens, and they are forming communities which are very different from dense forest, open forest, steppe, grassland, tundra. Then you have primary consumers, the herbivores, essentially here with large mammals, diversity of the mammals that could be present in Europe and in the hunting spectrum of Neanderthals, including cold species, ubiquistic species, and warmer species, here the Straxus elephant and the rhino, which is different from the mammals and the woolly rhino. So you have either the same species is present all the time, like horses and red deer, or the same functional species is present, proboscidean, but it might be a mammoth if it's cold, it might be an elephant if it's warm. And on top of that, what is interesting is that the predator spectrum is more or less the same all the time, with three big carnivores, the lion, wolf, and cave hyena, and of course the Neanderthal. The Neanderthal does not say that it is carnivore, but it's eating meat anyway, like the brown bear. So it's part of this group. So why use isotopes? So isotopes have the advantage of on three elements, so carbon, nitrogen, and oxygen. So I will talk essentially about carbon and nitrogen, oxygen just a little bit. And these three elements, of course, are essential for every animal. So carbon and nitrogen through the food, and oxygen through water, either drunk water or water from the food as well. And we are lucky that these elements have two stable isotopes with different masses. And the ratio of these two isotopes for carbon or for nitrogen or for oxygen change in nature according to some environmental parameters. And these environmental parameters can be predicted. And so we have a possibility to relate some abundances of stable isotopes, which are expressed as delta values, which is just a way for including a standard to make the values easier to work with. And for carbon, nitrogen, and oxygen, we have variations in nature, and these variations will be reflected by the animals according to their diet and the water they drink. As paleontologists or archaeologists, we have not the whole animal, but we have just the skeleton, essentially. In the skeleton, we have two types of molecules, or where we can measure it. One is bone collagen that we heard already before, and bone collagen contains carbon and nitrogen, so we can measure carbon-13 and nitrogen-15 abundances. And you have enamel bioapatite, so the mineral fraction of the bone, which is a calcium phosphate, but this calcium phosphate has the advantage to include 3–5% of carbonate as well. So in this carbonate fraction, you have carbon and oxygen isotopes that you can measure. I put enamel because of diagenesis, because of the modification of bone after death. Usually the carbonate in the bone is not reliable. It will change, and we cannot be sure that what we measure is really what the animals died with. So enamel, in contrast, is very stable, is highly mineralized, so it's sure that it will be reflecting the signal of the living animal. But as it was mentioned already by Bruce, the collagen and the tooth enamel have different time frames. They do not reflect the same part of the life of the individual. Bone collagen is remodeling all the time, so it's 10–20 years of lifetime, so it's an average, it's average seasonality, so it's really like an average of values recorded by the animal for many, many years. In contrast, tooth enamel, depending on how you sample, if you sample a very small part, you will have a snapshot of the life during the time when the tooth was growing, so which is essentially during infancy. So you don't have exactly the same signal in terms of what is reflex of the lifetime. Then, from these herbivores, you have carnivores, or different types of carnivores, and they will also record the carbon, nitrogen, and oxygen values of the prey, especially carbon, nitrogen, depending on what are the values in the prey. So how to prepare the samples and how to measure these values. So nowadays we need relatively small quantities of material. So for bone and dentin, in order to extract collagen, we need about 50 to 200 milligrams, so it's not very much. Of course, if there is collagen. And what we do to test if there is collagen before we take a piece, we drill just 5 milligrams. And with this 5 milligrams, we can measure nitrogen. And nitrogen will tell you if there is collagen or not, because if there is no nitrogen in the bone, you are sure you have no collagen. So it's not worth to take a bigger piece and to try collagen extraction, because it will not work anyway. So we have ways to screen the samples before we start really the extraction. The extraction is necessary because we cannot just run the whole bone. The whole bone is full of contaminants, so we have to make a preparation which eliminates minerals, contaminants, and so on. There are different ways, but the essential part is that at the end, we have a product that has the proper carbon, nitrogen content of collagen. So we have a device that will automatically prepare the sample for isotopic analysis. So it will burn the sample and then extract the CO2 and the nitrogen from the combustion and separate the elements, CO2 and nitrogen, and separate the isotopes and measure them. So with this device, we can run about 50 samples a day. So the longest part is preparation of the samples. But once they are prepared, they can run pretty fast. And we have a way to test for the quality of the collagen. So even if we have values, we can test after the preparation we have done if this value is correct or not. And we can discard values that are not based on well-preserved collagen. For the carbonate fraction, we need even less material, a few milligrams of tooth enamel. Then there is also a pretreatment to remove any exogenous carbonate, because of course carbonate is carbonate, so if you put a mixture of carbonate from the tooth and carbonate from the sediment, it will be measured at the same way. We eliminate what is left of organic material, and then it's a reaction that takes place with orthophosphoric acid, which is specific to carbonate, another way to purification. And there is also extraction of the CO2, and then measuring the carbon and oxygen isotopes and the percentage of carbonate, because there is a certain range of carbonate in tooth enamel. If you are below or above, it means that your enamel has been somehow modified. It's very rare, but it may happen. So we have also ways to test for the preservation. So for carbon, we have two sources of information, collagen and bioapatite. But because of the fractionation, so the changes that happen in these ratios of carbon in the different tissues of an animal, we have to correct the values to reflect the diet at the beginning, so the plants that were eaten by the herbivores. But we know how to do that because we have a lot of samples from modern animals. So we have the plants, we have the animals, they can be captive and be fed a certain type of plant, so we can measure everything. And there have been a lot of studies on that which allow us to develop some equations that will calculate this thing. So this is the isotopes of the ecosystem, so meaning the plants eaten by the herbivore, eaten by the carnivore, so we can tell where the plant food comes from for a given herbivore or carnivore if it's for the prey, and the same for carnivores. So this will be when I will give, sometimes we have sites which will have only collagen values, so we can just compare everything as collagen values. Sometimes it's all carbonate values, but sometimes it's a mix. And so if we want to compare carbonate and collagen values, we need to do that. So as mentioned before, the main reason for the difference in plants of carbon isotopes is the photosynthetic pathways, the C4 and C3 photosynthetic pathways. But we are in Europe under temperate or cold conditions, so we have only C3. And as mentioned before, the range is much smaller, but we have some variations. And these variations are big enough and consistent enough to be used as a tracer of the habitat and the type of food of different herbivores. But it's not just the plants, it's also how, in which environment they grow, what environmental factors are important, like the temperature, the nutrients, light intensity, and also water stress, saline stress. And for instance, we have in saline environments much higher carbon isotopes than in wet environment. Lichens are also interesting because they have a different isotopic signature as vascular plants. So for these reasons, we can we will see that we can distinguish the plants eaten by the herbivores. And as I said before, when we combine carbon and nitrogen isotopes, we can put some areas of a graph where the values of the plants from a forest or from a seafree grassland, and then the herbivores, so the meat of and then the carnivores eating that, and also the aquatic food webs. This is, of course, very simplified, and we will see that it's much more complicated than that, so we have to be careful. The preservation. So we are talking about Neanderthals. So Neanderthals in the classic range, they range from about stage 2, broadly speaking, and stage 5, so between 20,000 and 130,000 years ago. And in this time range, you see that the collagen, this one, is usually preserved until 20,000 years ago or even more, but when you reach 120, it starts to be a bit difficult. And this preservation does not just depend on the time, it also depends on the conditions. If it's cold, it's better. If it's warm, it's not so good. So like for DNA, you have some climatic conditions that are better or not so good for collagen preservation. For the apatite in enamel, it's no problem. We can even measure enamel apatite in dinosaurs. So here we are not in this chronological problem. Just an example to show how collagen might be preserved in a site. So a site in Belgium, SCLADINA cave, where we have a long sequence from the stage 3 to the stage 5. And we have bones more or less everywhere. So we can compare the content of collagen in bones from these layers compared to the content on modern bone. So it's a percentage of collagen compared to fresh bone. We start with 50% more or less, and then it declines up to 0 when we reach the layers below the MES5. And this is a site where collagen is very well preserved. Almost all the bones have collagen until there. So it's really a good context to go to collagen isotopes. So these are the sites essentially in Western and a bit Central Europe where collagen from Neanderthal bones have been extracted, or at least has been attempted to be extracted. And the results, so in this time range, compared to SCLADINA, they are listed here. So we have, in yellow, we have bones where the collagen, some collagen or something that, well, looks like collagen has been extracted, and the red where no collagen was present. So the nitrogen content was close to 0. And unfortunately, some very important sites with Neanderthals could not be used for this method because they are too old or the conditions were too harsh and collagen is not preserved. All of the others here, they yielded something. Now the question is this residue that we have extracted, is it really collagen? And so for this, we use the carbon and nitrogen content and ratios, which changes in very narrow limits in fresh bone collagen. And we use these limits as the area where the collagen, fossil collagen should be. If it's outside, it's not fossil collagen, it's something else. So all these areas which are not in this green part are no-go, so discard the values. So SCLADINA cave, for instance, all the values are here. So perfectly with like fresh collagen, so there is no problem here. Another site which is much younger in Sudan, 2,000 years only, but the values are all over the place and most of them are outside the range. So it's not a question of time, it's really a question of conditions. And when we do that for all these residues that were extracted from Neanderthal bones, we say that some of them are actually not good enough to be used for stable isotopes, even if maybe in the original publication they were claimed to be good enough, but actually new measurements have shown that they were not. And some of them, like this one, we don't have the data. This is a problem because some people publish their isotopic values, but they do not give the chemical composition of the collagen. So you cannot evaluate if the values are good or not. And this should not be happen, but unfortunately it still happens. So now we have still this ecosystem, and now we have some ways to link the trophic levels. So as it was said, the nitrogen 15 increases with trophic position, and also the type of habitat where the food comes from. So the herbivores come from, and the plants eaten by the herbivores come from. But we have also some range. I mean, it's not like we have one value for the plants, one value for the herbivores, one for the carnivores. We have quite a large range for each of the trophic system. Why? Well, because, so what we can do first is that we can use the Neanderthal bone, carbon and nitrogen isotopes, compared to the potential prey to calculate how many proportion of the prey were eaten. We can also compare the Neanderthal bone collagen isotopes with those of predators and see how they split the resources. Do they compete? Do they eat the same thing in the same proportions, or do they eat something different or different proportions of the same thing? And finally, and we have also most of the time, we have no Neanderthal bones available. We have only the fauna. But even with the fauna, which if we can relate it to the Neanderthal activity, if they have fauna with cut marks, we know they were eaten by the Neanderthals or manipulated by the Neanderthals. So they are potentially in their diet. We can look at the habitat of these animals that were hunted or collected by the Neanderthals. So one first thing is that most of the time, I mean, we'll see that most of the values I'm talking about will be from late Neanderthals. And so we are and also in France and Belgium. So this is essentially still the mammal steppe ecosystem. So let's have a look at the mammal steppe. And the herbivores in the mammal steppe, there is a feature that is really something typical of the mammal steppe from Europe to Alaska, across Siberia. It's the distribution of the main herbivores. So what is striking is that these herbivores, they have very different nitrogen isotope values. The range is from about 3 to 7, here 5 to 10. So we have already this 5 per mil difference between the highest and the lowest herbivore. So it's clear that it's not just herbivore, it's not one nitrogen values, it's a lot of range. And what is interesting is that the pattern is the same everywhere, especially the pattern for reindeer. Reindeer is always the highest carbon isotopic values. It's on the right side of the graph because they eat more lichen. And the mammals are always the highest in nitrogen 15 values because they eat more grass. So this is useful because then we can distinguish some type of prey based on their stable isotopic values. Why do they have these differences? We know that plants use different sources of nitrogen, and for instance, the plants with woody plants like trees and shrubs, they use nitrogen from mycorrhizae, so fungi that are in symbiosis with them, and grass use directly the nitrogen from the soil, so they have higher N15 values. And so grazers will have values higher than browsers. So it means that the terrestrial food web is not one food web. It's several food webs with different plants, different herbivores, and the values of these herbivores will be different from each other, and the carnivores eating them will be also different from each other. And as you can see here, for instance, a herbivore from this food web will have higher N15 values than a carnivore from this food web. So the nitrogen values of bulk collagen do not reflect the trophic level. They reflect the trophic level within the food web, but not globally if you mix different food webs, which is the case in Neanderthals. So let's take an example with Neanderthal and hyena from Saint-Césaire in France, one of the latest Neanderthal. And we have the typical herbivores and the average values of the Neanderthal and the average value of the hyenas. And as it was said before, the Neanderthals is higher in nitrogen 15 values than the hyenas. But it's not because it's more carnivores than the hyenas. It's because the nitrogen source is higher. And among the prey, you have quite a range. So here we have an area where the prey comes from, which is calculated from the difference between the collagen of the predator and the collagen of its prey. And the result should be somewhere here for the Neanderthal and somewhere here for the hyenas, where the possible prey range overlaps with the possible prey range of the predator. And of course, it's not the same. Here you have essentially these herbivores, so reindeer, horse, megaloceros. Here you have more these herbivores, so mammoth, rhino, and bison. And with mathematical models, we can make a Bayesian distribution of these. So it's not just one value, it's a range of values. And the most likely values would be here for the Neanderthal, for the mammoth range, but it's still possible to have more, but the probability is lower, and so on. So here what comes out is that mammoth is the highest in the diet, range around 30%, while reindeer is the lowest, close to 0, can be 0, but a bit more, and horses, rhinos, bison are higher. In Belgium, we have here the herbivores, the carnivores, and the Neanderthals. Again, we have Neanderthals above the hyenas and the lions and the wolf. So again, are they hyper-carnivore, more carnivores than the carnivores? Not. You have also the mammals high here. So when you calculate the proportion of the prey for Neanderthals, you find high proportion of mammals. Also, rhino is not bad. Reindeer is higher in this case, and horse and bison are low. So we have a possibility to evaluate. I would not say quantify, because these figures are, of course, based on the assumption that you have only this prey in the diet, which is probably wrong. But you can compare predators in the same site, you can compare Neanderthal and predator in the same site, and see if you can explain the differences by changing the proportions of the prey, which is the case. And so these late Neanderthals in Gouillet, it's P, in Saint-Césaire, they all have a high proportion of the mammals, and the others are lower. So this is something that comes out from these results. Of course, it's only 2 areas, 2 sites, late Neanderthals. So also something that is a limitation of this approach is that what you get is only the diet of the analysis of the samples you analyzed. You cannot generalize to everyone because it's just an individual measurement, but you can compare in a population how the variation happened. Do the specimens, so if I go back here the Neanderthals are really all together, the hyenas more or less as well, but the lions, for instance, are very different from one individual to another, which could indicate that the lions are more solitary hunters, So they hunt different things from one individual to another, while the humans and the hyenas are more, group players. So they are in populations that will do everything together and share the food and eat the same thing. So, high proportion of mammoths. So why is it interesting to hunt so much big animals? It's dangerous. But we have seen that the other carnivores do not really eat them. So there is a resource where you have no competitors if you can do it. And also these animals provide a lot of food. This is a calculation made by Cole where a group of 25 people could live 40 days on a mammoth carcass, 15 days on a rhino, 9 on a bison, 2 on a horse, and only 1/3 of a day on a saiga, just breakfast, not much. So it's, so of course it's an advantage to hunt big prey. And big prey, like mammals, they do not just provide meat. Of course, meat is important. Also, fat is important. These animals are very fatty. They also provide raw material for weapons, tools. I mean, not Neanderthals are doing that, as far as we know, but they could use the bones anyway, and also for different things that they need to do. So it's something we have, but we have to be careful, because how did we get to these conclusions that Neanderthals, or at least these Neanderthals, were eating a lot of mammals? We are assuming that the high N15 values can be only explained by Neanderthal meat. But we have a fish. We have seen that fish gives also values that could be similar to the values of the Neanderthals. Here is the food web with the Neanderthal food web with the mammals, and here it's a Mesolithic food web from France 8,000 years ago, and you see the fish are here. So quite similar to the mammals. So how can we be sure that it's not fish rather than mammals? Also, these collagen values give only the proportion of proteins in the diet. And so plants are more or less not possible to evaluate. Just a quick view of this graph where we see that meat has about 50% carbon, 14% nitrogen. Plant food, on average, of course there are some exceptions, but they have about the same, 40% carbon and only 1% nitrogen. So 15 times less nitrogen on average. And if you mix the diet of a pure herbivore and a pure carnivore, if you do a linear mixing, you get this. But if you take into account the concentration effect, so that there is much less nitrogen in the plant than in the meat, you see that immediately when you take 10% of meat in a 90% plant food, you get already a very close value from the carnivore. And with 50% of plant and 50% of meat, you have more or less the value that is indistinguishable from a carnivore. So you cannot tell how much plant could be eaten. Even if you get a value up there, you could still have easily 50% plant food that is not visible. So we are not telling they are only eating mammals. They get the proteins from the mammals, and besides, they may have a lot of plants that we don't see. So now it's coming a new method that is starting to be used to solve these problems. It's a single compound nitrogen isotopic composition of collagen amino acids. So collagen is made of chains of amino acid, about 1,000 of them, and each amino acid has a different story to tell. So traditionally, we measure the bulk. We measure everything. But among these amino acids, you have essential amino acids, which the body cannot make by itself, so they come automatically from the diet, and non-essential amino acids, which are synthesized by the body from other sources, other amino acids or other substrates. These essential amino acids, they do not change much from the diet to the predators, so from the plants, the herbivores, and the carnivores, either in the aquatic food web or in terrestrial food web. But a non-essential amino acid like glutamic acid changes a lot from one step to another at 8 per mil. And so it's possible to see the difference between a plant, a primary consumer, so herbivore, and a carnivore by comparing phenylalanine as an essential amino acid and glutamic acid as a non-essential amino acid. On top of that, you see that aquatic and terrestrial food webs are quite different. And it's also possible not only to tell the trophic position independently of the fauna around based on this method, but also to distinguish aquatic and terrestrial food webs. So this would be a way to tell apart fish and no fish. So the method is a bit more complicated than the collagen method. So first you have to cut the collagen fibers into amino acids, so this is not too difficult. And then you have to derivatize the amino acids and to send them into a gas chromatograph to separate the amino acids and send them one after the other into the mass spectrometer, but more or less automatically. And each amino acid will be measured separately for a sample. This is the ideal case. Of course, it's delicate to work on, and so there are not so many values available yet. But now, instead of having this kind of picture where you have different sources, different herbivores, different carnivores, you have this picture. So all the samples which are plants, they are different, but on this line, the herbivores, even if they eat different types of plants with different nitrogen values, they will end up on this line. So this will be the herbivores, and the carnivores will be up there. If you eat a mixture of plant and animal food, you will end up somewhere in between. And if you eat aquatic food, you will be somewhere here. So this is a better distinction between the different resources. Let's test it on bears. Bears are very interesting for stable isotopes of Neanderthals because they live in the same range. You have also, they are quite flexible in their diet, so you can test, and you have plenty of bones that you can test the methods on, while Neanderthals are more difficult to get. So here we have, and we have two species, one the cave bear, which is extinct but was essentially herbivore, and the brown bear, which is very omnivore. So among the cave bear here, so this is also from the same site, from Goyer, we have two values here which are very high in nitrogen 15, and I told you that they were herbivores, so how can they be high like that as herbivores? Well, they are the babies. And the babies, they drink the milk of the mother. They do that in winter when the mother is hibernating, so it's a stage where the blood physiology changes and it's using its reserves. So this explains this on the bulk collagen. The other ones, the adults, they are all low in nitrogen, but there is some variation. So the question is whether this variation reflects some different plants with different isotopic values, or if these ones are really herbivores and these ones are maybe a bit omnivore. And among the brown bears, we have a larger variability, which could be here herbivores and here more omnivores or even carnivores. And when we look at the results of amino acid analysis, we see that this line, the herbivore line, all the yellow points, including these ones, are there. So these are herbivores, even the babies plot as herbivores. And the blue ones, the brown bears, well, some of them are on the line of herbivory, even some with high N15 values. So it means that they were herbivores eating plants with high N15 values. And these ones, which have lower N15 values, are actually more omnivores than the other ones, because they eat, the food web they belong to has lower N15 values in the plants. So it really evaluates independently of plant nitrogen values where the animal fit into the omnivore range. And here are the carnivore line. So if it were pure carnivore, oops. And when you reflect all these bear values on a trophic position graph, so 2 is herbivore, 3 is pure carnivore with some uncertainty. So all the cave bears fall into the range of herbivores, some of the brown bears as well. Most of the brown bears are in the omnivore range. And here is another bear, which is from North America, extinct, and it was very carnivore in Alaska. So if it works with bears, why not for humans? So here are values of archaeological humans from different environments. A lot for Japan, because Yuji Naito, with whom we work, is from Japan, and he has been working a lot on Japan material. So here are sites from inland with sea-free plants. So everybody is between the purely herbivore and purely carnivore range, but not purely carnivore. They are omnivores. Here we have marine food eaters, like whale hunters, or people living in the coast and eating shellfish. And we have Mesolithic people from along the river near Paris. And there is a lot of fish in the site. And not surprisingly, they have high N15 values. And they have a position which shows that they are, well, in the range of the aquatic food eaters. So where are the Neanderthals? They are here. So they are not there. If they were eating some fish, they would shift here. But here they are well aligned along parallel to the pure carnivore line, but not on the pure carnivore line, so slightly below, and one of them just like modern humans from the Holocene. Here another type of graph where we see again that the marine food with high trophic position, so the whale eaters, the other marine food eaters, the freshwater eaters, and here the Neanderthals together with other terrestrial consumers. So there is no indication of significant aquatic food consumption. And I say significant aquatic food consumption. It doesn't mean that they don't eat the fish sometimes, but globally it doesn't represent much of their diet. And here we have a comparison between the fauna of Spee and in Belgium, where we have the values of bulk collagen with herbivores, the mammals up there, hyenas, Neanderthals, and now with isotopic values of amino acids. So all the herbivores are here, including the mammals. The only thing is that they eat plants with higher N15 values than the others, but the fractionation between the plants and their collagen is the same. And we have a wolf and the Neanderthals. The only thing that is not yet clear is why the hyenas are up there. They should be here. But hyenas, we cannot rule out an impact of scavenging. Maybe they eat not fresh meat, but decayed meat. And also maybe they eat each other. I mean, they eat also carcasses of carnivores, so they could increase above the level of carnivore. This is something we need to investigate further, test if it's also true for other hyenas in other sites, or if it's something specific to this site, we have to clarify. But anyway, the herbivores, the bears, the wolf, and the Neanderthals fit well. And the position of the Neanderthals compared to the pure carnivore line, so 2.8, means that they eat 20% of plant food. Okay, so, and we come to this reconstruction, which is very simplified, to show that, okay, Neanderthals eat mammals and rhinos a lot, but they eat also plants. So just to debunk this idea that they hunt or they eat only big animals' meat. And what is interesting is that the other predators eat something else, essentially, and even the predators among themselves, they have different choice of prey. For instance, lions are more focused on reindeers than the other predators, so probably to avoid too much competition among the predators. Now let's go to the topic of modern humans and Neanderthals, and where the modern humans more flexible in their diet than the Neanderthals. This is a graph taken from a PNAS paper from about 10 years ago, where the plot is just through time, Neanderthal nitrogen 15 values compared to early modern humans nitrogen 15 values, and with assumptions that the herbivores and N15 values did not change through time. And they are higher than these ones. So why should they be higher? Because they eat maybe fish. And so this is the key for the success. They could eat fish when the Neanderthals had to work hard for getting mammoths. But remember, we have the mammals also in the picture, so it's not, fish is not the only source of high N15 value food. These early modern humans had no fauna associated. They are just isolated humans that were dated by radiocarbon dating, and since you get collagen from the radiocarbon dating, if you have some leftover, you can measure the table isotopes. But you have no fauna, so you don't know how it's the baseline. And this is something we wanted to test, so we did a test in Dordogne, so in sites from the Mousterian, Châtel-Péronnien, early origination, and later origination. What is interesting is that when you combine all the values of the main prey of these people, so the horses, bison, and deer, you have a shift about 2 per million average, starting with the beginning of the proto-origination. And when you look at the values of these early modern humans compared to Neanderthals, they shift of the same value. And also the wolf shift the same way. So, of course, these values are not from the places where the modern humans have been measured, but, well, it's anyway sending a warning signal that before we tell, okay, it's a different diet, it could be also a different baseline. Now let's look at one of the best investigated early modern humans in Europe. It's a site, Buran Kaya III, which is in Crimea, so farther east, of course, but the advantage is that you have origination, lithic, you have human remains, and you have faunal assemblage. So you have everything in one place. So you can do the collagen, you can do the isotopes, and when you do it, you have this pattern. So the herbivores, also interesting, we can put the small, there are a lot of hair there, so we put the small mammals, and they have a lower N15 values than the rest. So you can also distinguish the consumption of small prey from big prey. Saiga antelopes are quite interesting because they are too high, higher in carbon than what we got usually. And this is probably because of their ecology. Saiga today, they also like to eat Chenopodiaceae in salt marsh and so on, and they have a signature that fits with these habits. Then we have mammals here, and we have some carnivores, and wolf and foxes are very widespread. And the humans up there. So the humans are very high in nitrogen 15. Values of Neanderthals were around 10, 11 per mil. Here we are 16, 17 per mil. So very high. The question is whether it's because of fish consumption or it's because of mammals consumption. Of course, mammals consumption, there is no mammals in the site except for some ivory bracelets. And this is a piece of the broken piece of the ivory bracelet that we can measure. But there is no mammoth bone in this place. So the zoologists said, no, it cannot be mammals. There is no mammals in the site. Yeah, but maybe there is a mammals a few kilometers away. And there is a paleontological site a few kilometers away with some mammals in it. So the dating is not so accurate, but anyway, it proves that the environment was not totally devoid of mammals, and it could be mammals. So to see if it was mammals or fish, we did the amino acids. And the amino acids show a clear pattern. So all the herbivores end up on this line, Saiga as well. The carnivores with very variable N15 values, they also end up close to the line, the foxes especially, with variable N15 values, but still consistent with the omnivorous, essentially carnivorous diet. And the wolf is on the line of a carnivore. And the humans, they are here. So they are high in nitrogen 15, not because they are high in trophic position in this direction if they were eating aquatic food, but because the plants of the prey they are eating, so probably the mammals, they were high as well. And here, the position of humans give 40% of plants, so slightly more than the Neanderthals from Spee. And here, a more complete graph where you see the position expected if it were aquatic, so fish, it would be here. And it's there, well between the terrestrial herbivore and carnivore lines. So mammals is confirmed, and fish is not coming into the picture. And this is a reconstruction of a diet of different prey on 3 different specimens of humans. You see the rabbit is really low, mammals is very high, and Saiga and the large herbivores are also there. So now we have this pattern. Bulk collagen, Neanderthals are lower in N15 values than the modern humans. But when we put them on the amino acid graph, well, first we can see that the Neanderthals we had are from Western Europe. The humans we had, they are the Crimean humans, but the other ones are from Oase Cave and Kostianki, so really East Europe. So it's not comparable directly. And the values we have for the amino acids, for both of them, they are in the omnivorous range, terrestrial range, a bit more meat versus plant in Neanderthals than in the humans, and no aquatic food. It's just the baseline that has changed. And what is interesting is that the modern humans have a bit more plants, but it doesn't mean automatically that humans were more, all modern humans were more herbivore or more, eating more plants than the Neanderthals. I mean, Crimea is warmer than Belgium, so it could be just an effect of the environment. And here we have, so the paradox, apparent paradox, is that these Neanderthals, which have lower N15 values than the early modern humans, they had more meat than the modern humans. So only this method could tell us the story. So here we have very simplified the contribution of mammals versus other prey for this Buran Kaya. We have also data from a slightly younger Gravettian human from Predmorsch. Also a lot of mammals, but not surprisingly, there are plenty of mammals bones. There are 1,000 individuals in the site of mammals. And the Neanderthals from Saint-Césaire and Goyer. And when we look at this, we see more or less the same pattern. A lot of mammals in all of these. So Neanderthals, early modern humans, they seem to eat the same thing. But these humans are from east, these ones are from the west. It would be nice to compare the Neanderthal and modern human in the same site. Now we can. So in Goyer Cave, we have Neanderthals, but we have also modern humans. Neanderthals are dated around 42,000 years, the Neanderthals 35,000, the modern humans 35,000 years on the same site. And we can see that the diet pattern is more or less the same. So it means that we have no big difference between late Neanderthals, early modern humans in one place, in one ecological context. So the modern humans have ecologically replaced the Neanderthals. We don't know if they came after the Neanderthals were gone, and so they use the niche, which is empty now, or if they came with the same niche and pushed the Neanderthal out. This we cannot say. It would be necessary to have really in the same level, but nobody has that. So if you have, please tell me. So they are doing the same thing. What happened here? Yeah, and here it's just to tell that the Neanderthals and the carnivores have very different diets, so we can also test the niche partitioning between these two, and Neanderthals, or also modern humans, are the only ones that get access to the mammals directly. And this is contributing to the debate, scavenging versus hunting. If it were scavenging, well, the hyenas would be the first to get to the mammals. And if it's so much mammals in the diet of a Neanderthal, it's probably hunting rather than scavenging. And here the same, only the humans have access to high mammals meat. The other carnivores have much less. Only the hyena have a bit more, the scavenger, because it may come after the Neanderthals to finish the carcasses of the mammals. So this is what we can do when we have Neanderthal bones and the fauna associated. First, be sure that the collagen we have is right. Then compare with the fauna. We can do the bulk collagen isotopes to reconstruct the protein sources, but also the single compound amino acid nitrogen isotope to see the trophic position and check for aquatic contribution eventually. Now, very quickly, because I have not much time, this is where the Neanderthals analyzed for bulk collagen isotopes come from. And this is the range of the Neanderthals. So we have only a very narrow view at the moment of this. And we have plenty of sites with fauna associated to Neanderthal industries. So now I will try to show you a few examples what we can do with these cases, which is the most common part. So first is I've said, okay, there's no fish in the diet of the Neanderthals in Goyer and Spee, but there are some sites like in Kudaro 3 in the Caucasus, Mousterian, where you have a lot of salmon bones in a layer associated to Mousterian. So it's interpreted as a Neanderthal eating fish. But you have also lions and bears in the site. So it's also possible that the lions or the bears eat the salmon and bring it to the cave instead of the humans. And so we did a reconstruction of the diet of the lion and the bear to check if they were eating salmon. When we do the bulk collagen here, carbon nitrogen, the bears are herbivores, they are cave bears, and the lion is a bit in between, so we cannot totally exclude some salmon in the diet. But we have, to sort out this, since the salmon is marine, it has a very different sulfur isotopic composition, and here there is no evidence whatsoever of a position between the terrestrial and the marine sulfur isotope. So it means that this lion was eating a terrestrial prey from this range, probably saiga or reindeer, and not salmon. So it doesn't mean that Neanderthals is automatically the salmon consumer, but it says that the lion and the bear were not. So it does not go against. Another example in southern France, southeastern France, so Baume Flandin, Eemian site, so interglacial. We have a temperate fauna with fallow deer, roe deer, red deer, wild boar. It's dated to the Eemian. And the idea is to see what they did. We have been talking about Neanderthals in the mammal steppe. Now, what are they doing in the Mediterranean area when it's warmer? And here we have essentially large herbivores. And what is interesting is that we have bones with cut marks, essentially from some species, and the bones with cut marks and bone they are from red deer and roe deer, and bones with bite marks of carnivores, and they are from bison, horse, and reindeer. So we can make a sketch of a trophic system where the deer are mostly consumed by the Neanderthals and the others by the carnivores. And when they have, where have they been hunting? So if we base our reconstruction of the habitat of these prey on modern analogs, we would be tempted to put the bison, horse, and reindeer in an open landscape and fallow deer and roe deer in a forest landscape, while the red deer is more difficult because they are everywhere. But actually, all of them are everywhere. If you look carefully, you find also population of horses in forest or bison in forest and fallow deer in the open. So it's difficult to tell where they really lived in this time. So we can use the isotopes for that. And here are the carbon isotopic values of different species of herbivores. Remember, the ones here are the ones hunted by the Neanderthals. And they have some, so here is the line between forest and open landscape, and we have a few samples which are at the limit or even in the forest, while the rest is in the open, while the species hunted by the carnivores are more in the open. So maybe there is a difference in exploitation of habitat. It's not so many values, but if you have more data, more samples, you can maybe make something bigger and interesting. Pear, again, we have heard about this site 1 hour ago, and here we have a site with which is middle slope to the Rhone Valley here, and the plateau here. And we did this time carbon and oxygen isotopes. And we have a very nice bimodal distribution of the fauna. Some species have low carbon and oxygen isotopes, which is consistent to a valley habitat because it's wet. The river comes from the Alps with low oxygen isotopic values, while the plateau is a karstic plateau, so very dry. And this would explain the higher C13 and higher O18 values. And this is where the species would be based on these isotopic values. What is interesting is that the zooarchaeologists were putting the horse on the plateau because the horse needs to run, and the red deer on the valley because the red deer lives in the forest, but it's the opposite. So, and in this case, we could even analyze the carbon isotopes of the Neanderthal enamel. And we have two levels with no evidence of environmental change. The fauna is the same. We have carnivores, wolf and hyena here. The wolf are mostly hunting on the plateau, the hyena in the valley. Here the lion is also in the valley. And the Neanderthals, what is interesting is that in this case, they are in the valley, at least when the tooth was forming, so as children. And here they are more in the plateau. So it seems that between these two layers, they have changed their behavior, although we have no evidence of environmental change. So they just decided to do something else. And finally, examples of what happens through time and geographically between Mediterranean and northern latitudes. So we have here SCLADINA again with these layers from MES 3 and 4, MES 5. So here we have carbon isotopic values of the ecosystem of herbivores, carnivores. Everything is in the open, not surprisingly. And the Emian, many values are in the forest, especially for red deer, fallow deer, even reindeer, horses. And here the values in the forest are actually from, not from woolly rhino, but from the forest rhino, and here from the straight-tusked elephant and the mammoth. So we can see the different habitats of the prey, and also some carnivores also feed on prey from the forest. Now, southwestern France, cold, Emian, not differences. I mean, these places, they are really did not change much between cold and warm conditions. So no wonder they are refugia. And here, when you compare the Emian in Belgium, dark green, and in southern France, you see the forest is much deeper, much more dense in the north than in the south. And now I played around with this figure where I tried to put some reconstruction of on SCLADINA in the north. So you have steppe tundra or forest with openings, of course. It's not all forest, but nevertheless, it's really densely forested. If the animals go to the forest and eat a lot of forest plants, they cannot they prefer to go in the open, so they have no choice. They have to be in the forest. So what we can do when we have no Neanderthal bones, but we want to understand their ecology, well, with carbon, we can tell where the habitat, what was the habitat of the different prey and how they use the landscape. It can be also compared the prey of the predators and the prey of the Neanderthals to see if they hunt in the same territories or not. And if you have a sequence with different environments, you can see how things change through time. Now, of course, we are not finished. We are just beginning, and we know we have more work to do. Of course, we would need more Neanderthals because the more we have, the better. I mean, for the moment, it's restricted. We need to do more of this single compound amino acid analysis because it's a new technique. It's in development. So we just get the machine installed last week. So now we can start working on new specimens that we have already and maybe more. And of course, extending the geographical and chronological range of these studies would be great. And we are well aware that we are missing some prey, like birds. We are not taking birds into account at the moment. Also, small game is just starting to be included, but not much. So, yeah, it needs to be improved. So I'm really thankful to my team, and especially these 3 researchers who have been essential in the work that has been presented. The support of archaeologists and anthropologists and all the collaborators of these studies, financial support, the conference for inviting me, and you. Thank you.